Hello and welcome to part 2 of the plasma membrane video series. Last video we talked about just the structures of the plasma membrane and how that plasma membrane is able to form a barrier because of those hydrophobic tails on the phospholipids. Well, now this video we're going to talk about this essential question. How does that plasma membrane allow some things to pass through while keeping other things out? And that is a very important quality of the plasma membrane. It's how the plasma membrane is able to help the cell maintain homeostasis. As you guys know, there are probably things that we need to get into the cell, like food and oxygen, and there's things that we're going to need to get out, like waste products and carbon dioxide. So today we're going to look at, in this video, how that actually happens. First, I just want to remind you guys of the structures that we see. If we were to zoom in on that plasma membrane, the structures that we might see, the phospholipids, they are creating that phospholipid bilayer, and as a result, we have this hydrophobic region, and we also have the hydrophilic region. And remember that hydrophobic means water cannot be around that area, so no water through this area. It also means that this area is nonpolar. So it's not just water that it wants to keep out, it's all nonpolar molecules. And that's going to take us, oh, yes, we need to remember that this is a fluid mosaic model. Fluid meaning that everything can move around. Mosaic meaning that there's lots of stuff other than the phospholipids there, like the proteins, which we're going to see how those play an important role in transport. So let's take a look next at what is actually able to pass through this membrane. First, I want to give you guys some terms to look at. The first one is semi-permeable. Semi means some. If we do our root words here, semi means some. Permeable means pass. Another word for this is selectively permeable. Okay, so it's going to be able to select what can move through and what can't. And another just kind of reminder here, back when we talked about water and we talked about solvents and solutes, we talked about a term like dissolves like. And this is going to help you guys kind of orient yourself into an understanding of what can move through and what cannot move through the plasma membrane. So first, let's look over here. This structure here, with you guys might recognize these little purple structures as being a phospholipid. We have our hydrophobic region, where our tails are, and then our hydrophilic region, touching the extracellular space, which is going to be the light blue, and our intracellular space, or the cytoplasmic space, which is going to be represented with this sort of purplish blue here, okay? So what can pass through? Well, anything that is hydrophobic and small is going to be able to pass right through or diffuse right through the plasma membrane. Okay, so things like oxygen, carbon dioxide, nitrogen, and some of our small steroids are going to be able to pass right through. Other things that can pass through are small, and I mean very small, polar molecules like water, glycerol, urea, and ethanol. And we're going to look at some particular things with water here in a little bit. But yes, they can very, very small, so they can just kind of sneak past that hydrophobic region. It may take a while, kind of like they're having to sneak through a barrier, so it may take a while, but they can pass through. Larger molecules like glucose is not going to be able to pass through. Another type of molecules, anything with a charge on it, like ions, are not going to be able to pass through because they're not going to be able to go through that non-charged or non-polar region. So how do we get things, or how do things actually pass through that region? And that's going to take us to two main types of transport. We call this cellular transport. And the two main types are going to be passive transport, which does not require any energy, and active transport, which does require energy. We're going to start by talking about passive transport. The first type of passive transport we're going to talk about is simple diffusion. And if you guys remember back from when we talked about the Skittles and how if I put a Skittle in water or a food coloring in water or anything like that, what's going to happen is it's going to start, and I'll draw a quick little picture up here in the corner. So if this were a glass of water and I were to drop in some molecules, they're going to start very highly concentrated in one area, but because of random kinetic motion, these molecules are going to want to spread out. So over time, these molecules are going to spread out throughout my entire glass. Well, the same thing happens here, but only this time we're going to talk about it spreading out across the membrane. So if you take a look at my picture here, I have extracellular space and I have intracellular space. So this would be the inside of my cell. And right now on the outside, I have a very high concentration of oxygen. On the inside, I don't have any oxygen at the moment. Because there is a difference in concentration across this barrier, across the plasma membrane, this is called a concentration gradient. And a concentration gradient is needed in order for diffusion to occur. Okay? And anytime we do have a concentration gradient, what we can expect to happen is that those molecules, or the solute, oxygen in this case, is going to move down its concentration gradient. In other words, it's going to go from a high concentration, which is on this side, to a low concentration, which is on this side. And we can see that happen. Oxygen is going to diffuse across the membrane, and it's going to continue to do this until we reach equilibrium. In other words, until we have an equal amount of oxygen on the outside of the cell and an equal amount of oxygen on the inside of the cell. Okay? Now, something to point out is that oxygen is going to continue to move here, but it's going to be moving at a steady rate back and forth, and that's going to help maintain that equilibrium. So another case of this is, what about carbon dioxide? Can it move out of the cell? And the answer is yes. We have a concentration gradient here. So we have carbon dioxide in. We need to get it out of the cell. We have more inside than we do outside, so there is a concentration gradient present. It is going to move down its concentration gradient, which means it's going to move out of the cell, like so. And it's going to do that until we reach equilibrium. And again, it's important to pass out there was a net movement out of the cell, but once it reaches that equilibrium, it's going to continue to move, but it's going to do it equally. So then this takes us to the question of, well, I know that I need to get glucose into my cells because I need that for cellular respiration, but if glucose is too big to pass through the cellular membrane, how does it get in? And this is actually a simpler answer than what you think. It's still going to pass via diffusion, and there is still going to be a concentration gradient. Notice that I have more glucose outside than I do inside. And it's still going to move down its concentration gradient. The only difference here is that I have to have a channel protein in order for it to pass through. So basically, this protein is going to come in, and it's going to act as a transport carrier, meaning it's going to be like a little road through the membrane, allowing glucose to pass. And again, that's going to happen until we reach equilibrium. Our last little thing that we need to talk about with passive transport is osmosis. And this is a form of simple diffusion or facilitated diffusion, depending on the case. But this is going to be specifically talking about water. Now, what happens here is we still have a concentration gradient. It's still going to move down its concentration gradient, but we're going to add in something called aquaporins. And aquaporins are these little small openings or pores all along the plasma membrane. And this is what water is going to be able to travel through. Because if you guys remember, I have this hydrophobic region. So how is water going to get across that? And if you remember me mentioning before, water is really small. So sometimes it can sneak through, but that's going to take a little while for that to happen. So instead, we have discovered that the cell has aquaporins in it, which are these tiny pores. And water is still going to move from a high concentration of water to a low concentration of water. So in this case, the water is going to move into the cell until we reach equilibrium. Another way to look at osmosis is that water is going to move towards a high concentration of solute. So in this case, I have these little red triangles representing solute, maybe sodium. And they're outside of the cell is a higher concentration of solute than I do have inside of the cell. Well, what we need to realize here is that that sodium cannot pass through the membrane. So it's going to try, but it's not going to be able to pass through. So if it's not able to pass, water still would like to move towards and even out the total amount of molecules on both sides. So what water is going to do is move towards that high concentration of solute. Since I have more of the sodium outside, water is going to travel out until we have reached equilibrium. So some terms that we need to know when we talk about osmosis are the terms hypertonic, isotonic, and hypotonic. Hypertonic is going to be where I have more solutes outside of the cell. So water, again, is going to move towards those high concentration of solutes. It's going to move out of the cell. Isotonic is where it's even for both water concentration and solute concentration. So water is just going to move back and forth evenly. Hypotonic is where I have more concentration of solutes inside of the cell than I do outside of the cell, so water is going to move in. Well, something very interesting happens here. If water is moving outside of the cell, and remember my cytoplasm is made up of a lot of water, so if water is moving outside of the cell, my cell size is actually going to shrink. It's going to lose mass and shrink. If it's moving in and out equally, in our isotonic situation, my cell size is going to remain the same, and this is what a red blood cell should look like. If water is going to move into the cell, my cell size is going to gain mass, and it's going to swell. And this is also a problem because it can swell so much that it bursts and breaks the cell. Same thing happens with my plant cells, only I have an extra built-in layer of protection here with my cell wall. So what happens here is in a hypertonic situation, I'm losing water, so my cell is going to shrink away from the cell wall. In an isotonic situation, that cell membrane is going to be pressed right up against the cell wall, and I'm not losing or gaining water, so I'm good. And in a hypotonic situation, I'm going to gain water, and as a result, I'm going to put pressure on that cell wall. And this is actually a happy cell right here. It likes to be in this situation. So while this hypotonic situation is not so good for an animal cell, it's a very great situation for a plant cell. We're going to finish up our conversation talking about active transport, which is going to require ATP. So this is where things are going to actually, I still have a concentration gradient, have more molecules inside than I do outside, but this time I'm going to move against the concentration gradient. And this is going to require energy in the form of ATP. So what happens is ATP comes in, loses a phosphate, becomes ADP, and now I can move in a molecule against its concentration gradient. And this is very important because there are some things that we need a high concentration of inside of the cell versus outside of the cell. Another form of active transport is when I need to move really big molecules in, or several at a time. And this is also going to require a high amount of energy, but basically what happens is that my membrane itself will actually create an infold. Those molecules will travel into that infold, the membrane will pinch off, and now I have a vesicle inside of my cell with molecules in it. And whenever this is going into the cell, we call this endocytosis. So this would be when my molecules are too big to just simply diffuse through, and I need several of them at a time. Okay, active transport in the other direction is going to be exocytosis. So I have a vesicle, it's going to fuse with the membrane, and it's going to release those molecules out into the extracellular space. And again, this is going to require a massive amount of ATP for energy. And this is going to wrap up our discussion about the plasma membrane. I just wanted to point out to you guys that not only is this happening on the plasma membrane that separates the inside of the cell from the outside of the cell, but all of our membrane-bound organelles, this kind of transport is happening across them as well. And this is how a lot of our cellular functions occur within inside of the cell. For example, you can see this happening here in the Golgi apparatus, is we have a vesicle going through exocytosis, And this vesicle will actually travel to the plasma membrane and then go through exocytosis again to exit the cell. So this happens again all throughout the cell. We see passive transport occurring across the membrane of the mitochondria, and we'll get into more detail about that when we talk about cellular respiration. But that's all I have for you. Thanks again for watching. I hope you enjoyed and learned something from this video, and have a great day.